If you like the presentation, I'm Ayllon-Benitez. In the other case, I am Patricia Tebo. The revolution of sequencing technologies increased the interest of study gene set. Here, for example, we are interested by analysis of the impact the genes into a given disease. So we can compare these two population samples in order to quantify the genes. And then by using method like clustering method, we can group them by similar expression. Here we are interested to know the biological information for the gene set. So for that we use the gene annotation, but the number of annotation can be huge for ungiven gene set. So for that we use a statistical enrichment analysis tool in order to compare the gene set with a reference to use a statistical method to obtain a list of enrichment annotation terms. But there are two main limitations. The first one, this method only highlights the very well annotated genes, discarding information of the very poorly annotated genes, obtaining at last loss of information because we have loss of annotated genes. And the other issue is this method considers the relation between the genes and the annotation without considering the structure of the annotation that can be stored, providing redundancy information. And we need to use post-treatment analysis to minimize this information. I propose a new method in order to use semantic similarity measure or a graph theory with three main objectives. The first one is to reduce drastically the number of terms. The second is maximize the gene coverage and propose an interactive visualization facilities in order to make easier the interpretation. We use the gene ontology in order to retrieve the annotation for each gene, and then using the structure of the gene ontology, we reduce the information, the inappropriate information, like redundancy and incompleteness. Then, for a given gene set, we have a list of gene ontology terms that we use we use the semantic similarity measure in order to obtain a matrix of similarity gene ontology terms. And that we use this matrix as input in clustering method, especially hierarchical clustering method, to obtain an hierarchy of similar terms. And then by using a score to obtain the optimal number of clusters, we have the cluster of similar terms. Then for each cluster, we re-explore the gene ontology in order to provide one or few representative terms for each clustering. And then we have a list of representative terms. Also, we reduce the first step in order to remove the potential redundancy and incompleteness that we can create by this method, keeping only the most pertinent information. And at last step, we used a method that used algorithm problematics that's called gene set cover problem in order to obtain the number of representative terms as synthetic, keeping the number of genes. At last, with the representative term, we propose also an interactive visualization in order to make easier the interpretation of the result. This visualization, we use the representative terms in order to obtain all information with the gene ontology graph, but we transform this graph into a tree, keeping the most important relation between the terms. For that, we combine two tree visualization, and circular tree map and identity tree, and we provide interaction facilities in order to explore this result. We have here some examples of the possible interaction that we can hear for the visualization. We propose an herb and package that include the method proposed before and also the visualization. So thank you very much. If you want to use JSON, you can use by this Git repository. And if you want to contact me, don't hesitate for any question. Thank you very much.
Hi, I'm Victor Granda, and I work at the Center of Ecological and Forestry Research in Barcelona. and today I will talk about how we build the first global database of subflow measurements, the SolutionNet database. Subflow is a proxy of plant transpiration, meaning the water lost by the plants to the atmosphere, and it's really important to understand how the plants will adapt and will cope with the different climate change scenarios. We have compiled data from more than 200 sites and 34 countries, consisting of continuous measurements of individual plants. This will provide with data and the tools for scientists and modelers to help them unravel the global patterns and drivers of plant transpiration. SolutionNet is completely open. It's an open science project with collaborating with scientists around the globe. It's completely open source, mostly R, but we use other open source tools. And it's open data, with all the data available in a Zenodor repository that you have there. So how we did this? We needed to design a completely reproducible workflow to start from the raw data from the researchers to the final clean data product. In R, everything is a package. So we designed all the steps of the quality checks and the data flow as functions of the SolutionNet QC1 package. In this way, we can automatize almost all the process and also test that we are doing that we want to do. We can check for metadata quality, like coordinates, species names, etc., or also data itself correctness, outliers, ranges, and also missing data. All these steps generates automatic R Markdown reports and logs that can be used to inform the data contributors of the process and also to check wherever if something went wrong. Of course, we aim to complete automatization, but always some manual steps are always needed. For this, we design Shiny apps that record all the user interaction with the Shiny app and generate R scripts that, again, in case of data loss or server failure, and believe me, this happens, we can use this to completely and automatically reproduce from the beginning to the end and the final, to get the final data. When we got the data, we build the database and upload it to the Zenodor repository. So we have the data, but we need tools to inspect, analyze, and visualize the data. Here enters the SolutionNet package, already in CRAN. This package introduces a new S4 class, the SVN data class, that can be used to inspect the sites, and, for example, selecting only the sites that we want to work with, for example, Mediterranean biome sites, and also it introduced methods to aggregate the data. We have continuous daily data, and we can aggregate to daily, monthly, or even yearly values. And also, even selecting what parts of the day we want to aggregate, midday, pre-dawn, daylight, or nighttime. This is really important because pre-dawn and midday are, in plant physiology, are really important moments for the plant. Of course, we can modify the data objects in a tidyverse style. We can mutate, we can subset, and even we can create new data from new experiments to be able to compare our data to the data present in the SolutionNet database. And finally, we also develop simple visualization methods using ggplot that can be used to generate the plots, and also the user can modify and customize and theme the plots in order to use it in publication or wherever. So that's it, and I want to thank the Spanish government for their funding and the CREAF for providing the servers and the work infrastructure. And this is our webpage and our Twitter in case that you want to know more about us. So that's it, and thank you very much.
Hello, everybody. Indeed, I'm Fabiola Gamba, and I'm a senior statistician within the Janssen pharmaceutical companies of Johnson & Johnson. So indeed, what I would like to talk today is actually a brief overview of my PhD research, if I'm able on time, because I only have 5 minutes. So the title indeed is Bayesian sequential integration within a preclinical PKPD modeling framework using R-SAM package. And this is a joint work with Tom Jacobs, Jan Seroy and Helena Guess from Janssen Pharmaceutica and Crystal Fass from Hasselt University. So now actually I said it too fast. Okay. So first of all, I would like to give you a background. So in a previous paper, we developed a novel PKPD model to assess the synergy resulting from the co-administration of two compounds. Here you can see the model. I can't be too much detailed about this model, but in general, it is a nonlinear hierarchical model that is kind of difficult to fit. Also, the interesting part is that we have 11 different trials that were collected over different time points. So the idea is to apply this model on these 11 trials that are integrated sequentially so that the posteriors coming from one trial are used in order to determine the hyperparameters of the prior or the next trial. So we kind of make an integration chain where the information is constantly updated. And this is a kind of appealing approach, but we have a challenge that we have a very complex nonlinear hierarchical model to be applied on small data in each integration steps. So we could have practical identifiability issues. So the aim of my research is to study the different factors that are influencing these results, and we use R-SAM package. So STAN, as you know, is a very cool Bayesian software that allows a very efficient estimation, and R-SAM is indeed the interface to STAN. So the first aspect that we checked was the prior specification, as you can see in this slide. So what we did was to rerun the analysis each time with a different, indeed, prior precision. So a different prior standard deviation, as you can see in these graphs. And what you can see is that the parameter correlations are increasing with a decreasing prior precision. So this highly correlated parameter structure gets reflected in the parameter estimates that result bias. So a first take-home message from this case study is that whenever possible, it is better to borrow the information from previous trials whenever possible. So we can really avoid these compensations among parameters. A second aspect was the choice of the random effect. So what we did was to rerun this time the analysis with a different allocation of the random effect. And what we noticed is that if we were placing the random effect on a parameter that was highly correlated with others, like indeed a K-out, I can't enter into detail about K-out, but this indeed model was giving biased posterior predictions, whereas a parameter that was easier to estimate, that was not highly correlated with others, was giving much better predictions. And this is because we have overcompensation, again, among parameters. So we should try to restrict the variance, indeed, of the parameters that are very high correlated. This was another aspect that was the design of the sequential integration. So what we did is actually, first of all, integrate the trials where each of them was assessing only one dose combination. Not because we wanted, but just because the scientists came to us with these trials already designed. This is, of course, not a very good design because, of course, we only have one dose combination. We don't have enough information for the estimation of all the parameters. So we kind of accumulated bias that propagates till the end of the sequence. So for that reason, if you can see the first graph, you see indeed that the predictions are not very good. Whereas if we really have trials that are assessing all the possible dose combinations, we have much better results, as you can see in the second graph. So this is because indeed we have identifiability issues. So what I did later on was to do a simulation study. You can see the slide very soon, yes. So in this simulation study, what we did was to compare the Bayesian pooling, so just pooling the trials all together with the Bayesian sequential integration again, using different models. So we start with a linear model, very simple, and then we continue with two different nonlinear models, just to see if we can confirm our results. And so indeed what we notice is that if we have a linear model, everything is fine between the two techniques. We can really use the Bayesian sequential integration without problems. But again, we had the confirmation that if we have nonlinear models, and we have a hierarchical structure, and we use an informative prior distribution, we see a bias for the Bayesian sequential integration. So that, again, confirms the fact that this is an appealing approach in drug development. We kind of tempted to use that, but we should really be careful in the nonlinear hierarchical models, because we should take care of different modeling aspects. And with this, I would like to finish. Thank you very much for your attention. And if you want to ask me any questions.
So good morning, everybody. I'm Boris Hejblum, and today I'm going to present you our work on Vici, which is a Shiny app for accurate estimation of vaccine-induced cellular immunogenicity with a bivariate modeling. And this is joint work with Edouard Lhomme, Rodolphe Thiebaud, and Laura Richert, which are who are all part of our research team at the University of Bordeaux. So this work takes place in the context of the clinical development of vaccine, where immunogenicity evaluation is a key step, especially when we don't know the correlates of protection, which happens, for instance, in the development of HIV vaccines. And in that case, we are going to measure a lot of immunological markers at once. And in particular, here we're going to focus on intracellular cytokine staining, which is going to measure the production of cytokines by the cells. And how does that work? We get samples from the participants, and for each participant, we're going to do ex vivo stimulation of the cells. So you got one sample that's going to undergo no stimulation, and other sample from the same patient that are going to be stimulated with different antigens. And once this is done in the laboratory, the samples are sent in a flow cytometer, and you get ICS data. So the way those ICS data are currently analyzed is quite simple. What people, and when I say people, I mean immunologists, are doing is they subtract the response from the non-stimulated sample from the antigen-stimulated samples. And the idea behind that is to focus directly on the antigen-specific response. But because you do this very simple subtraction, you get type 1 error increase, which you don't want, obviously, and as well as bias, and also low statistical power in some situations. So there's a need for a better statistical approach. And that's why we developed this bivariate linear modeling. So this slide is a bit heavy, and I won't have time to go into the detail of the model, but it's not very new nor very sophisticated. The idea is to model the two dependent variables between the non-stimulated response and the stimulated response within the same individual. And I'm sorry, but because I work with medical doctors who do a little bit of programmation, but they use SAS, so I had to put some SAS code here. But the important point is that we can also, of course, do this in R. So this is great. As statisticians, we're happy. We can estimate a model that allows us to better estimate what we care about, which is the vaccine effect on the stimulated response. But immunologists are usually not trained in programming, and they cannot use what we've done as is. And that's why we developed a Shiny user interface. It's called Vici, and you can try it out because it's deployed at the URL here on the far right. I'm going to walk you through the app now. And the first step is to input your data in the app, and that's what you get. You get a dynamic view of your data. So this is real data. I'm going to show you real results, so that's why I've blurred the line. But then the next step is you need to select which variables are going to be the one in your model. So what is the response variable? What is the antigen stimulation variable? And then you can feed the model, and you get two graphical outputs, which are displayed here. So because I had to send my slides to Natalie 3 weeks ago, here you have a heat map with only one line, but the app has been updated, and now you get a heat map with more lines. And you also get numerical table of the results, as well as this box plots here, which allow you to see all your comparison. So to do this work, we rely on a lot of different packages. The dependencies are listed here, namely DT, ggplot2, ggpub-R, Magritar, NLME for the statistical model, and of course, Shiny. So for the interested statistician, we also have additional information such as dynamic formulas of the estimated model and a few extra numerical results on such as the AIC or the estimated variance. So in conclusion, we propose an improved statistical model for analyzing ICS data. We have implemented a Shiny app, which is still under development, but it's deployed and available as is. And it's available also as an R package on GitHub, and it's going to be on Crown soon as we reach stable. Thank you.
Hello, everyone. I am Deniz. I am from Turkey, Başkent University. I have a very long title, but the main subject is demand management. We used some market basket analysis to see test patterns. Before talk, I want to talk about shortly about me. My background is in medical medicine, but I am using R for 4 years. I was trying to analyze this in Excel, and now I am improved with R. This is my second useR participation, and I am very happy to talk here. Before explaining the application, I want to explain our clinical laboratory workflow. We are very strict workflow. The clinicians order tests, from the patients, urine, blood, any other samples are taken. It's transported to the laboratory, and we perform analysis. The most critical step can be the test ordering step is very critical because there are lots of tests, over 3,000 biochemistry or endocrine or microbiology tests are possible. So sometimes it is difficult to select these tests, and there is an increased cost problems in medicine and in laboratories. So we must manage these test requests. There is a concept that demand management in clinical laboratory. We can achieve this with market basket analysis. As you know, market basket analysis can be done with association rule mining. It's a pattern mining. For example, if you eat hamburger and fries, then you eat cola. This is some simple example. For laboratory, it can be for a renal failure patient. If you order creatine, you also want to see blood urine. We can use a priori algorithm for this. This is applied in lots of fields, but there is a rare item problem in a priori algorithm, and this is very important in clinical laboratory tests. I will talk about it. About our app, we use these packages. The main packages, Ayllon and Ayllon-Vici package, supports for association analysis. We used our university tests, 1 year test order. This is our main and the only secret. We developed a shiny app because the medical doctors aren't familiar with statistics, as the other speaker says. So this is very simple secret to use. There is only one button to generate rules. You can filter patient type, age group, or tests because the rare item problem. So you can filter more frequent tests or least tests, and after generating rules, we can also visualize this. In the slide, we can see 3 different groups. The bleeding test, 2 tests are in there. Acute coronary syndrome related test, troponin and CK-MB, and the infertile test can be grouped as in the graphic. So the clinicians or the management can see the patterns. Again, about the rare item problem, to overcome this problem, we also order tests as frequency, so we can limit the search parameters. It can help us to determine more patterns. We are planning to use multiple support thresholds. There's an article about that. MS a priori is more suitable for rare item problem. We are also planning to database integration and more user-friendly reporting for this tool. Managing test requests is very important now in clinical chemistry, and we are proud to provide this tool to clinicians and to laboratory management. Thanks for your attention.